Hi, this video is the first part of chapter 12 on muscle physiology. So we're going to be looking at characteristics and organization of skeletal muscle mostly. We're going to leave the other as cardiac and smooth muscle for just a very small, short discussion. And we're going to see how muscle fiber contracts when we're talking about skeletal muscle. First of all, what are the characteristics of muscle? If you want to be a good muscle, what do you have to do? You have to be able to contract. So contractility is very important. Extensibility, it has to be able to stretch passively. So when you have one muscle contracting, you have the opposing muscle stretching passively. That is extensibility. It has to have elasticity, which means after it's contracted, it has to recoil back to its resting length. It has to be able to relax. And it has to be excitable. It has to respond to stimulation. And that's going to be from a neuron that we've been talking about. So a neuron is going to come, neuromuscular junction is going to excite that muscle. This chapter comes with its own terminology, so I want to go over it. When we talk about a muscle cell now, we can also refer to it as a muscle fiber, which is very confusing because you think of fibers as elastic fibers or collagen fibers, but in this case, when you say muscle fiber, you mean one cell. When we refer to the cell membrane, you might remember this from anatomy. It is called a sarcolemma. Sarco means flesh, so it's the membrane of this flesh. And cytoplasm now becomes sarcoplasm, so the inside of that cell. And we have a modified endoplasmic reticulum, and it's called the sarcoplasmic reticulum, and that's where we store calcium in our muscles. Okay? Very important that you refer to this always, that you remember the different terminology. Just a quick reminder there of skeletal muscle, cardiac muscle here that's shorter and has the intercalated discs, so the gap junctions, and smooth muscle where we don't tend to see the striations. So again, we're going to be concentrating mostly on skeletal muscle. How is it organized? We're going to start from big to small. So if you have one big whole muscle that you're thinking of, pick any. Within it, you're going to have fascicles that are bound together by fascia. So think of fascia, I think of it as a tape, like duct tape. If you were to take some sticks and duct tape them together, then you have this stronger structure. So a whole muscle are fascicles that are bound together by some sort of tape, by some fascia. And I'll show you in pictures. The fascicle then, what is that? It's a bundle of muscle fibers, which you remember are cells. So if I get a bunch of cells and I wrap this tape around it, this fascia around it, I'm going to form a fascicle. If I get many fascicles and I wrap those around, I'm going to have an entire muscle. The cells themselves, the fibers, if you look deeper into them, they actually are loaded with these myofibrils. So it doesn't look like Joe's cell when you look inside, and it just has a nucleus and some ribosomes. It's going to look different. It's going to have these myofibrils, muscle fibers. And these are made up of two very different proteins. And we're going to call for now one is the thick one protein and the other is the thin protein. And they actually are forming filaments. The thick filament is particularly which protein? It's called myosin. The thin filament is which protein? It's actin. So if we build back up, we have actin and myosin in these myofibrils. You have bundles of myofibrils are inside a cell. Bundles of cells make a fascicle. Bundles of fascicles make a whole muscle. Okay? Hopefully you're getting the picture here. In a diagram, there we go. You have here a muscle cell. This one. the muscle cell, then you have many muscle cells bound by fascia, you have a fascicle. And then if you have lots of fascicles, all these bigger dots, right? Fascicle, many fascicles together are going to make the entire skeletal muscle. So that's the breakdown. And hopefully you remember some of this from anatomy. A muscle fiber, reminder, is a single cell. It has many nuclei because embryologically they form separately and fuse together, and they end up with many nuclei. But this that you're looking at is just one cell. Now let's take that one cell and look inside. So we're dissecting that one cell. Here would be one of the nuclei of that cell. This is the sarcolemma, and I've cut inside, and I'm looking at this array of parallel lines here. These are the myofibrils. Okay, so we have myofibrils, and making up that myofibril that you see here is a thick filament and a thin filament. So this arrangement of muscle is really different from like a skin cell, right? And the reason we have all these filaments and myofibrils is because this cell has to do something really important, and that is contract. And if it's going to contract, it needs this hardware essentially to be able to do that and not look like a skin cell. What we want to look at is what the arrangement of the thick filament and the thin filament look like. If I backtrack one slide, you see them kind of in lines like this. And the thin filament you see more red colored here, and the thick one you see more purple, and they all seem to be nicely parallel in lines. So it's a sarcomere, by definition, is a repeating bundle of alternating thick and thin filaments that are sitting there in parallel. That's a sarcomere. So in my diagram here, the orange would be the thick filament, and the black would be the thin filament. Thick, thin, arranged in this manner, and then sort of branching out to this end and ending on this zigzag line. Zigzag line. So from this zigzag line to this zigzag line, I have one sarcomere. You have thousands of these per cell. Depending on how big the muscle is, it'll have more or fewer of them. But it gives that muscle a striped look, like you saw when you did anatomy. And you see these striations. And what you're seeing is a darker area here is just one fat line, and then less dark here. And then darker here, fat line, and then less dark. So you see these lines. In this diagram, it's easier to see how everything with purple would look darker, everything without purple would look lighter. Okay? So, we name this orientation. So there's two bands. The darker one of the areas is called a band, and that's dark. So dark has the letter A in it, so that's the A band. That's how you're going to remember. And the lighter one of the two has an I in it. That's the light. Okay, so the I band is the one that's the lighter one, for you to remember which is which. And then this zigzag line is a Z disc. Remember that from Z disc to Z disc is one sarcomere. The only other one I want you to remember is, I think that's it. Yeah. So written out there for you, you have the A band, entire width of the thick filament, the I band, which is the thin filament only, and the Z disc, which are going to tell you which is the beginning and the end of one particular sarcomere. All right. What we want to look at, by looking at all this anatomy, we want to understand the sliding filament theory. In other words, how does muscle shorten? And this was a mystery for many years with physiologists. They couldn't figure out if a muscle was kind of like a coil, like a spring that would sort of spring out and spring back, and that's how it lengthened and shortened. Or it was more like a rubber band, that it was actually able to stretch. Something about those cells made them stretch and then retract. And this was somewhat of a mystery of how is it that a muscle can go from being longer to being shorter and then being longer again. Because a skin cell can't do that, right? A liver cell can't do that. It's these particular skeletal muscle cells that are able to do this. So that is called the sliding filament theory when they actually figured it out. And what they figured out was that it was an overlap of actin and myosin that was going to be the trick of the anatomy in order to really understand how muscle shortens. I'm going to stop this video here, and the next one's going to be specific. I'm going to start with this slide on the sliding filament theory. Thanks.